again, just a quick outline of what we're going to cover. So we're going to look at the structure of the tissues involved, namely the xylem and phloem. We are going to look at the mechanisms by which water is being transported, namely the transpiration stream, basically, and also we're going to do some experiments on how we measure its rate and what factors affect it. And then we're also going to look at translocation. We're going to sort of hone in on active loading and the evidence that supports the mass flow and the mass flow itself and the evidence that supports it, and also the evidence for the cohesion tension theory. So let's dive right in. First of all, the structure of the xylem and phloem. First, a sort of a little bit of a general description of the plant's transport systems. And the note that if you're AQA, you don't need to know about how the xylem and phloem are arranged in a plant, but all the other exam boards you do. So very similar to animal exchange surfaces, right? Plants, like animals, need to be able to exchange gases and nutrients with their environment. For multicellular plants, the rate of diffusion is not efficient enough. So there are dedicated transport systems. Gas exchange, which only takes place in the leaves, does not need a dedicated transport system because the leaves have adaptations, which is basically their shape, that they're flat and thin, that maximize the surface area to volume ratio. So no transport needed for gases. Other substances, though, that are taken in, basically the substances that are taken in by the roots, as well as substances that are made in the leaf, namely glucose, but also amino acids and lipids, they need to be, they're produced, either produced in one part of the plant or absorbed in one part of the plant, so they need to be distributed to the rest of it. Now, a little bit of sort of structure of the xylem and phloem. So, of course, xylem transports only water and nutrients, or it's actually a little bit more correct, guys, to say, instead of nutrients, to say minerals, because nutrients can mean organic nutrients such as glucose and amino acids, whereas we know that the substances absorbed by the roots are actually water and inorganic minerals. So just make sure that you distinguish inorganic minerals. And of course, the sap, which is a mixture of water and sucrose, is transported up and down by the phloem. So the two vascular tissues absorb different things. This little line here is a cross section through a leaf. And leaf. And it basically shows that we have this kind of central portion here, which is the central vein or midrib of the leaf. And those little dots here are the vascular bundles in the leaf. Basically little bundles of xylem and phloem. Bundles. If we were to take a cross section, so this here is a cross section of a leaf. If we were to take a cross section of the stem here, we would see that the xylem and phloem are kind of like arranged around the sort of periphery. And if we were to take a cross section from a root, we would notice that actually the xylem is kind of like in the center here as a kind of like a cross, and the phloem is around it. So just kind of like be aware of that. Let's now look at the structure of the xylem and phloem individually, separately. So xylem vessels, and their kind of like distinguishing feature from phloem cells, is that they are dead cells. How does that happen? When the plant is still developing from a little sort of seed that it starts to grow, initially at some point, of course, of the life cycle of the plant, the xylem cells are alive. And as the plant matures and the xylem matures, these cells basically become dead. How? By losing, so you can see here that there are no end plates between them, there are no borders between them. So they basically form, there's no end membranes or cell wall, it's broken down to form a single hollow tube. So this here is a single hollow tube. Which allows for an uninterrupted flow of water from the roots to the leaves. Sorry, I should have made the arrow. It's very misleading. That's bad of me. I should have made the arrow that way because the direction of flow is from the roots to the leaves. They also have another adaptation, again, thinking of those questions that ask us to link structure to function. I'm just going to do it in another color here. They have another adaptation, which is that they've got a type of polysaccharide called lignin, which is what these sort of spiral lines are here, this and this. This is just to kind of indicate that the lignin can have a spiral or a circles kind of structure. And the lignin is basically a type of polysaccharide that waterproofs and strengthens the xylem. So as the xylem vessels mature and the cells lose their endpoints from each other, lignin is also deposited around the xylem vessel. Let's have a look at a typical question about xylem. So a student is investigating the rate of water movement through the xylem vessels in plants, and they find that 2 mL of water travels through the plant in 116 minutes. Calculate the rate of water movement throughout the xylem in liters per hour. Note the units. Give your answer in standard form. Also note instruction about the unit. So let's break down again our different sections of the question. So context first is describing to us the investigation of the student. So basically the volume and the time given, which we're going to use to calculate rate. The command word is that it's a numerical response, it's calculate. And my direction are the units that I'm going to use. So I'm being given information in mils and minutes, but I have a unit conversion to do because they want me to quote it in liters per hour. So take a quick minute to see how good you do with unit conversions, basically. Okay, so let's first of all do the conversions of the units before we calculate the rate. So we have mils that we have to convert to liters first. So how will we convert 2 mils into liters? Going from the smaller unit to the bigger unit, we divide, and the factor between liters and milliliters is a factor of 1,000. So effectively 2 mils are 0.002 liters. So I divide 2 by 1,000. Essentially, I'm just going to write here, divide by 1,000. Then I have to convert the minutes to hour. And of course, I know that 60 minutes is 1 hour. So how do I convert 160 minutes into an hour? I basically divide them by 60. So 160 minutes over 60 hours. So in order to find out the rate, of course, I have to divide the amount of water that travels in the time that it took to travel. So what I will do is I've got my 0.002 mils of water that traveled over 160 over 60. So if I solve all of that, that gives me 0.00075 liters per hour. I've got one extra step to do, which is that I have to convert my answer to standard form, because that's what I'm asked to do, which is standard form is used when we have too many zeros, right? So that gives me 7.5 times 10 to the minus 4 liters per hour. Okay. Now, let's look at the structure of the phloem in a bit more detail. So we said that the phloem is used to transport assimilates. What are assimilates? Assimilate is basically a term to mean the organic nutrients that a plant is able to make for itself by photosynthesis. So it's basically glucose, amino acids. It could also be lipids. And the direction of flow, in contrast to the xylem, which is upwards only, roots to leaves, the direction of flow in the phloem can be either way. It can be up or down. And we're going to see exactly why that is in a minute. So we said sugars and amino acids are assimilates. And of course, most of the assimilates are produced in the leaves. Why do we say most? Well, because there can be a little bit of photosynthesis in other green parts of the plant, such as the stem or branches, but mostly the leaves. But they're required all over the plant. They're required on, as we're going to see in a minute, on tissues that don't have access to sunlight at all, such as roots. And we're going to have a little bit of a look in a bit more detail at exactly the types of cells that make up the phloem, but it's basically made up of two types of cells, namely the sieve tube elements and the companion cells. The sieve tube elements, which are these ones here, sieve tube elements. Elements, just, it's a cell, right? So one element is one cell. They are lined up end to end, and they are connected by sieve plates. So these here are the sieve plates. What are sieve plates? Basically, they are the end bits of the cell, so where one cell would separate from the next, but they are perforated. So that's another point of structural contrast with the xylem. In the xylem, the end plates are completely disintegrated. In the sieve tube elements, they are just perforated. So that basically does facilitate the flow of water, but they're not as absent as in the xylem. What are other adaptations to maximize the flow? They have little cytoplasm, and they have also lost their nucleus and many of their organelles to maximize space for sap. Remember that sap is basically water and assimilates, right? Let me just write this here for you guys. Sap is water plus assimilates, which in turn are sucrose and amino acids. What do the companion cells do? Well, they basically accommodate for the structural adaptations that the phloem cells have done, right? So they have many organelles and a nucleus, particularly many mitochondria, so that they can basically provide energy to the sieve tube elements. And they also help us, we're going to see in a minute, with loading and unloading the sucrose, the main component of sap, into sieve tube elements, into and out of sieve tube elements. They also have, you know, a nucleus and a endoplasmic reticulum and ribosomes for protein synthesis, essentially. So these here are my companion cells on the right. And there are also some perforations. They have a direct communication, the companion cells, to the sieve tube elements. These particular sort of little communication points here between the two cells, are called plasmodesmata. Plasmodesmata, one word. So plasmodesmata are basically the way that neighboring plant cells can sort of link up to each other and exchange substances. Let's have a look at an example question. So plants contain two types of transport tissue, xylem and phloem. The xylem transports water and the phloem transports assimilates such as sucrose. Not quite sure why this particular question says that the xylem transports only water, because clearly it also transports mineral ions. A bit of a peculiar question. Explain how the structure of xylem vessels and sieve tube elements relate to their function. Again, guys, super classic question relating structural adaptations to function. I'm going to leave you some time to do this question, but before, let's break it down. So our context here is this sentence. It tells us that we're looking at xylem, phloem, and their function. Our command word is, of course, to explain, which means to give reasons for how the structure relates to function. And finally, my direction is to only focus on the xylem vessels and the sieve tube elements, so basically not companion cells. Okay. So, see how well you can do that. Okay. Let's have a look at the answer together. So in order to be able to explain how their structures differ and relate to their function, we need to actually also do a bit of describing in this explain question. So how do we describe the structure? So xylem vessels are lignified, that is one mark right there, which waterproofs them and also strengthens them, preventing them from collapsing under negative pressure. Don't worry too much about what this negative pressure means. We're going to see it in a little bit more detail in a minute. It basically refers to the idea that effectively water is being sucked up in a way in the xylem vessels, and that kind of like draws the walls of the xylem vessel inwards. We're going to see it in more detail in a minute, so don't worry about it too much now. But we've linked the structure, lignification, to what it does. It waterproofs them and it strengthens them. The end walls of the xylem vessels are broken down. That's another feature, structural feature. And there is no cytoplasm or organelles. That's another mark there. And what does this do? It allows the xylem vessels to form long, hollow tubes, which basically maximizes water transport. It is essential for transporting water in a continuous, unbroken stream. That's another mark. Sieve tube elements are connected by sieve plates. That's a structural feature, which create a long tube allowing the flow of sap. That's what they enable the phloem to do. Relating structure to function. Space is also maximized by reduced cytoplasm and few organelles. So again, a structural feature, reduced cytoplasm and few organelles. And what do? They maximize space. Okay. Make sure that you don't mention any structural feature without qualifying it with what it does for the function of the tissue. Let's now actually go and look at the functions of xylem and phloem, and namely, how do they transport water? So we're going to break down, so the transpiration stream, which we're going to look at the overview of, is the net movement of water from the roots, as we said, to the leaves. But in order to understand how it works, we have to start our study, our journey from the leaf, what happens at the leaves. And we have to be able to understand that it's as important as it is for plants to absorb water through the roots, it's as important to lose that water through the leaves, for that water to evaporate. So basically, water evaporates into the air spaces between these spongy mesophyll cells. So a little bit of a revision on your structure of your leaf. So we've got here, upper epidermis, waxy cuticle. Here are our palisade mesophyll cells right here. And here are our spongy mesophyll cells. And as the word suggests, they've got air gaps between them. If you remember, those air gaps also facilitate gas exchange, because they facilitate the movement of carbon dioxide from the stoma. This down here is the stoma. Stoma, or singular. Stomata, the plural. Again, for those that you might not know, stoma means mouth in Greek. So they're essentially the leaf's mouth, because that's where gas exchange takes place from. Here we have our vascular bundle, which will include both xylem and phloem vessels. However, here in this particular example, we're going to focus on the xylem vessels here. So what happens to water then? What is the path that water takes? It's going to, so first step is that water evaporates from the xylem vessels that is being carried into the leaf, it goes into the cells of the spongy mesophyll, and then let me just do this in another color so you can see a bit more clearly. It will evaporate. So here is my right 10. Here is my sort of step number 1, that water evaporates here. Now, be careful of one detail in terminology here. A lot of students are tempted to say that the evaporation of water is from the inside of the leaf to the outside, to the atmosphere. No. Evaporation technically means water going from liquid to a gaseous state. And technically, since these here are air gaps, water has already gone through from a liquid to a gaseous state. So evaporation is right here into the spongy mesophyll cells. This is a common mistake. Second step is that water then exits the leaf through the stomata down a water vapor potential. Water vapor, right? It exists as water vapor in here, and it exists as water vapor out here in the atmosphere. So there's a water vapor potential, a gradient, meaning what? A higher concentration of vapor inside the leaf than outside in the atmosphere. Finally, so we call the loss of water from leaves and other aerial parts of the plant is called transpiration. So the combination of all of these things that we have, that we've just described, right? Evaporation and then diffusion, basically, of water vapor out of the cell. Both those events together are called transpiration. Okay. What's the difference between the term transpiration and the term transpiration stream? The term transpiration stream involves the entire flow of movement of water. Let me just do this in blue. From the roots to the leaves. That whole thing is the transpiration stream. However, if the word transpiration alone means what is happening at the leaves, the combination of evaporation and diffusion of water vapor out of the leaf. So water first is lost by the leaves, and that causes a negative pressure within the xylem. And this negative pressure is what is called the transpiration pull. So the fact that we create a negative pressure means that water will be sucked up eventually, it'll be pulled from the roots. So therefore it's drawn up the xylem from the roots to the leaves under tension. Okay, keep that word tension because we're going to see towards the end of this session what the cohesion tension theory is and how it explains the transpiration stream. So again, don't worry too much about the negative pressure. And so the reason why water can be drawn up the xylem in the first place is because of these fundamental properties of water that you have learned back in the chapter when you were learning about water and its properties, which are the adhesion and cohesion. Adhesion and cohesion, guys, is basically just another name for the intermolecular forces, especially for those of you taking chemistry, the intermolecular forces between water molecules. So cohesion is basically the forces between water molecules, which are the intermolecular bonds or the hydrogen bonds that you've learned between water molecules. And the adhesion forces are the forces that develop between water molecules and the walls of the container, in this case, of course, the xylem vessels. Forces between water molecules and xylem vessels. Adhesion forces are basically, if you look at a glass of water and you can see that the water forms like a little bit of a meniscus, that's because of the adhesion forces of the water to its walls of its container. The adhesion and cohesion forces mean that the stream of water is in one continuous or unbroken column, as we say. If you're in Edexcel A, you don't need to know about adhesion, but you do need to know about cohesion. So as we say, the entire flow through the xylem is what is known as the transpiration stream. Okay. Let's now see what is happening at the other end of things. So we saw what's happening at the leaves to deplete water from the plant. Let's look at what's happening at the roots to replace that water that's being lost. So water enters the roots by osmosis. Never get tempted to say that water moves by any other mechanism, right? Water, osmosis, always. from a high water potential to a low water potential. Yeah, that's how osmosis works. Water moves from high to low water potential. There are three pathways that water can take to move from the soil to the root and then eventually to the xylem vessel, which remember, the xylem vessel, remember, if we took a cross section of the root, sits right at the center of the root, right? Whereas the root hair cell is here at the surface of the root. And so water effectively has to travel from the soil all the way to the xylem vessel here at the center of my root. So what is the path that it can take to do that? So we've got three different paths. If you're AQA and Edexcel A, you don't need to know the names of the different pathways. OCR and Edexcel B, you do. So we have, it's quite important that I can use the right colors here. So we have, if we look at these green arrows here, we have what is known as the apoplast pathway. So in green arrows. What is the apoplast pathway? It is basically water moving through the, what are these? The cell walls. Cell walls, remember, are made of cellulose, and cellulose is an incredibly hydrophilic molecule, meaning that water can sort of really easily diffuse through it. It's basically what happens if you take a piece of paper and you dip one corner of it, one end of it in water, you'll be able to see the water like kind of absorbing up. What's the second type of transport? The rest of the two types of transport are actually quite similar. And we've got the symplast pathway in red. Symplast pathway in red. And then we've got the vacuolar pathway in blue. So what does that mean? It means that water actually enters the cell, whereas in the apoplast pathway, it hasn't even entered the cell at all, right? It hasn't even passed the cell membrane. It's literally just traveling outside across the cell walls. Symplast and vacuolar means that the water has entered the cell. And symplast just simply means that it's traveling through the cytoplasm. And vacuolar means that it's also crossing the vacuoles inside the cell. So similar in that respect. We have an adaptation called the Casparian strip, which is found usually at some point. Let me just do it in red so you can see it. It's usually found, so if this is my root hair cell, it's usually found somewhere here, like at the end of this pathway. And what it does is that the Casparian strip is made of a waxy substance. And I know that waxy substances don't allow water to move through them. And so what it basically effectively does is that it forces the water to quit the apoplast pathway and to force it to travel intracellularly, which is important to be able to control its movement more. Let's have a look at an example question. So the xylem vessels are responsible for transporting water throughout the plant. Water is drawn at the roots and supplied to the leaves through the transpiration stream. Describe what happens to the transpiration stream if the xylem vessels are broken. So again, you're going to have a go at this, but first, context, the xylem and the transpiration stream right here. My command is describe, which basically means to, state or outline with no explanation needed. And my direction is to focus on the effect on the transpiration stream only, not the plant as a whole. So with that in mind, have a go. Let's look at the answer together. Cutting the xylem vessels will allow air into the xylem, effectively disrupting the transpiration stream and breaking the cohesion forces between the water molecules. So as we said, the cohesion and adhesion forces have to be in a continuum in order to allow the uninterrupted movement of water up the xylem, which is effectively moving against gravity, right? So 1 mark goes for saying what's going to happen to the xylem vessel. It's going to disrupt the transpiration stream. And the 2nd mark goes for explaining what is going to happen to the cohesive forces between water molecules. Okay, let's move to the next part then, which is what are the experimental techniques that we use to investigate the rate of transpiration? And also, what are the factors that can affect it? So the setup, the experimental setup, and I believe there's a little bit of overlap between this and even your GCSE. So you kind of learned that in a more simple level in your GCSE. Setup is called a potometer. Poto in Greek means to drink. So effectively you're measuring how quickly the plant is drinking the water. Now, let's kind of like break down the setup here. Why do we set it up like that, first of all? Like theoretically, we could be measuring the amount lost, the amount of water vapor lost from the leaves. But technically, this would be next to impossible. You'd have to somehow, because it comes out in very, very small droplets, you'd have to somehow collect it and then measure its volume. So it's much more straightforward to actually measure the amount of water being taken up. So how do we do that? Well, effectively, we replace the roots in the soil with just a direct water source. So we take a plant and we cut off just before the roots. We cut the shoot. We cut it underwater and we generally assemble the entire apparatus underwater because it's important that no air bubbles penetrate any part of this apparatus. We may also do other things such as, so it's important that we have no air bubbles here in any part. We also kind of like, sometimes you put a little bit of Vaseline wherever you've got connecting bits of tubes, again, to avoid any air bubbles getting in there. And we're going to see, because it's important that we have the one air bubble that we use. We also cut the shoot underwater so that no air bubbles enter the xylem vessels here, because again, it's important that we have a single column of water going up the xylem. We have here a reservoir, which is just used to top up water so that we can repeat the experiment many times. And we connect the whole setup to a sort of narrow tube here that has a ruler behind it, so I can measure the distance. I introduce a single air bubble here. That's why it's important I don't have any other air bubbles anywhere in my setup, because I just want the single air bubble here. As the plant is absorbing water through transpiration, this air bubble is going to move from right to left. I can record the distance that it travels, and I know the diameter of this tube, so I can also calculate the volume of water. So the 2 pi r squared times the distance to calculate the volume. Yeah, that's it. Another caution that I can do before I start my experiment is to pat dry the leaves before I start my experiment, to ensure that there are no leaves, to ensure that there's no water on them. What are the factors that I have to control? Like with most experiments, the control factors are going to be basically anything else that can affect the outcome of my experiment. So I use this entire setup to be able to investigate one factor at a time. This is how a proper experiment is set up. You only test the effect of one factor at a time. So what are the possible factors that can affect my rate of transpiration? Light intensity. The higher the light intensity, the higher my rate of transpiration, because the rate of photosynthesis is going to be faster. So therefore, the stomata are going to be open for longer to facilitate gas exchange, and therefore more water is going to be lost from the leaves by transpiration. Second one is the temperature. The higher the temperature, also the higher the rate of transpiration, because the rate of evaporation from the leaves, transpiration is going to be higher. The effect of humidity, relative humidity, so basically the humidity just outside the leaves. The higher the humidity, the lower my transpiration rate. Why? Because there's not going to be as big a gradient of water vapor between the inside of the leaf and the spongy mesophyll spaces and the outside. So the lower the gradient, the slower the diffusion of water vapor out of the leaf. And finally, air movement. So the higher the air movement, the faster, again, my transpiration rate is going to be, because as soon as a water vapor diffuses out of the leaf, it's being blown away by the wind, effectively keeping that gradient, that concentration gradient of air vapor steep. So humidity makes it less steep, air movement makes it more steep. And finally, availability of water. So the more water the plant has access to from its roots, the more it can allow faster transpiration to take place. Let's have a look at an example question, quantitative data analysis question. So a student was using a potometer to investigate the impact of total leaf area on the rate of transpiration. His results are shown in the table. So number of leaves on plant that represent the total leaf area, obviously, right? And the mean rate of bubble movement on the potometer, of course. Describe and explain what is shown by the student's results. So again, before you guys have a go, let's look at, let's break down our context. So our context here is basically this entire table, right? The data. My command is that my command words are, of course, describe and explain, right? Here's my command words. But how exactly will I utilize them here? Well, I'm going to use the figures to describe, for the describe portion, and I'm going to use my knowledge, my understanding of transpiration to give reasons. And then the direction is to focus on the results and giving the reasons from our own knowledge. So have a go at writing this answer. let's look at the answer together. So first, the describe portion, right? And as we said before, for my describe portion, I'm going to describe the general trend of what I'm seeing, followed by quoting a set of values to justify the observation that I gave. So mark number one, as the rate of leaves on the plant increases, the rate of bubble movement through the potometer also increases. So just by looking at the numbers here, I can see number of leaves increasing, rate of bubble increasing. Easy, right? As an observation. So that's my one mark. For example, just literally pick two data points and quote them. For example, with 0 leaves, the bubble moves at 6 units, but with 8 leaves, it moves at 95 units. That's it. Then the rate of transpiration increases with the number of leaves as there are more stomata. So now we go to the explain part. So now we're going to use our knowledge to explain why this happens. There are more stomata for water vapor to diffuse out of. Note, to diffuse, not to evaporate out of, right? And therefore the rate of water uptake by the roots is also increased. Because effectively the evaporation from the leaves drives the uptake of water, which is why a lot of times if a plant loses its leaves, it's not going to be able to absorb water from the soil. Okay, let's move to the next part, which is translocation through the phloem. Again, specification points, make sure you're super familiar with them. So in order to understand, before we kind of like look at translocation as a whole process, we have to be able to understand how the assimilates find themselves into the phloem, into the sap in the first place, right? And we said we know that photosynthesis creates glucose. The plant doesn't actually transport its sugars in the form of glucose, rather it dimerizes it into sucrose. What do I mean by that? The glucose is a single sugar. And remember from your chapter on carbohydrates that you were learning about disaccharides. Sucrose is a disaccharide. And there's several so the plant basically like links up a glucose, sorry, with a glucose and fructose, and that makes sucrose. And there's some advantages of transporting the sugars in the form of sucrose instead of glucose. Sucrose, by virtue of being a disaccharide, is less osmotically active. And so it's also more efficient because you're transporting 2 sugars at a time. So let's break down what happens to the loading. So basically, sucrose is loaded into sieve tube elements using active co-transport. We're going to see exactly what that is. If we kind of like zoom in, let me just move this window out of the way. If we zoom in to what is happening at this membrane, right? So this membrane is this membrane, which is the membrane between the sieve tube element and the companion cell. Step number 1 is that hydrogen ions are actively transported out of the companion cells. Active transport. Obviously, this here is a carrier protein. Carrier protein. Because remember, only carrier proteins, not channel proteins, are involved in active transport. Okay, fine. So what does that do? Sorry, there was something with the microphones. What does that do? What does this active transport of hydrogen ions outside of the cell do? It will create a concentration gradient, won't it? It'll create a concentration gradient of more hydrogen ions out here, out of the companion cells. Okay. Whenever we have a concentration gradient, what is going to happen? those ions, or whatever molecules, are going to have the tendency to diffuse down their concentration gradient. So those ions are eventually, let me just use a different color here to illustrate. Those ions are eventually going to diffuse back into the companion cells down. So I'm just going to say diffuse. They're ions, so they have to diffuse through an aqueous channel. So basically this here is a channel protein. It's actually a carrier protein. Sorry about that, guys. It's a carrier protein. Okay. However, so they're going to diffuse down their concentration gradient. Concentration gradient. However, this carrier protein is not a usual carrier protein. It's rather a co-transporter protein. A co-transporter, which utilizes the concentration gradient that the hydrogen ions are flowing down to co-transport.
Can you hear me?
To co-transport, sorry, there was a little bit thing with the microphones again. Co-transport sucrose in there. If you've done your revision, you know, you'll remember that there are many examples of these co-transporters, and those of you that are in year 13, you'll even remember them from your kidney. There's co-transporters in the intestine. So step number 1, hydrogen ions pumped out against the concentration gradient by active transport creates a concentration gradient of hydrogen ions outside. When these hydrogen ions diffuse down their concentration gradient, they do so via a co-transporter, which will also transport sucrose into the cell. So this is the active loading of sucrose into the companion cells, right? So here's my sucrose. Here's my sucrose into the companion cell. That means what? That means that now the sucrose concentration is going to build up into the companion cell. Where is the sucrose coming from? It's coming from the palisade mesophyll cells where photosynthesis is taking place, right? So sucrose concentration now builds up in the companion cells, and now it's going to simply diffuse down its concentration gradient into the sieve tube elements. Okay, so let me just write like the numbers here so that you can relate the diagram to the process. So process number 1 here, hydrogen ions out. Process number 2 here, hydrogen ions and sucrose going back in. Process number 3, sucrose here, sucrose diffusing from the companion cell into the sieve tube element. Okay, right. Example question. Companion cells actively load sucrose into the phloem tissue using the mechanism of co-transport. Explain why co-transport is a type of secondary active transport. We haven't explicitly learned the term secondary active transport, right? So we have to kind of like explain in this particular context. So before you guys have a go, our context is this. Oops, sorry, I just managed to zoom in excessively. How did I do? Pardon my technological challenges here. So, context. The fact that we're dealing with co-transport in companion cells. My command, the fact that it's explain, which basically means give reasons. And finally, my direction is to focus on what determines secondary active transport. So take a quick minute to think about the answer for yourselves. Okay. So the energy needed to move sucrose against its concentration gradient into companion cells is coupled with the diffusion of hydrogen ions down their concentration gradient. That is basically what active cotransport is, right? So that's one mark. However, the concentration gradient of hydrogen ions is first set up by actively transporting hydrogen ions against their concentration gradient using ATP hydrolysis as an energy source. That's the second mark, right? So basically, I'm explaining all terms, right? Secondary active transport. Why active transport? Because ATP is needed to set up the hydrogen ion concentration gradient in the first place. Why secondary? Because one concentration gradient is coupled with the transport of the other molecule. Okay. Now, let's look at mass flow, mass transport in the phloem. And let's define what a source and sink is. So the source is a little bit more straightforward, I would say, because it's basically where the assimilates are being produced, which is where the leaf, really. The leaves are sources. Remember from the structure of the leaf, from the cross-section across the leaf, that you've got your palisade mesophyll cells, which of course do photosynthesis. And then there's the vascular bundle inside the leaf, right? So the assimilates are made in the palisade mesophyll cells, and then they're loaded into the phloem. So the source is always the leaf. Now, what are the sinks, though? The sinks are the location where the assimilates are used and or stored. So basically, they're taken out of the phloem and they're put into the sinks. What are some examples of sinks? Well, the roots. Let me just change color to make it a bit more obvious. The roots, for one, right? The root is a sink. Doesn't have access to sunlight, cannot photosynthesize, has to rely on a supply of nutrients, of assimilates from mass transport through the phloem. Often the roots might have something like a potato tuber being formed. Potato or tuber, they're called collectively, like the root vegetables, which will store a big amount of sucrose that gets converted into starch, right? So that is also an important sink. A growing flower can also be a sink because it needs a lot of nutrients to develop. And so the movement of assimilates from source to sink is basically what we define as translocation. Quick mention of a possible trap that the potato tuber, for example, depending on the stage in the life of the plant, it can act as a sink, but if you take a plant, or a seed can act as a sink, but if you take a plant and plant it, so if you just like plant a potato that sprouts and makes roots and stem, at that stage the potato can be a source. Because the plant is still a seed, it can't photosynthesize. Don't worry too much about that. That it might be like, it's rare. It might be a sort of tricky question, but focus on the basics. Okay, so what are the stages of mass flow? Basically of mass flow, of translocation. So the word mass flow and the word translocation are not mutually exclusive. Sorry, they're not interchangeable, I meant to say. The word translocation, as we said, is simply the flow of assimilates from source to sink. Mass flow is a much bigger, sort of broader process that involves both the phloem and the xylem together. And it's a theory or a process that was suggested, described by scientists relatively recently. So let's break it down into steps. So step number one, I'm just going to do them in slightly different colors here so you guys can. Step number one, the loading of assimilates into the phloem at source, which we already saw. It's all that stuff that we said with the hydrogen ions and the co-transport. That leads to sucrose or amino acids as well, right? Being loaded into the sieve tube elements here. So here is my loading of my assimilates, number one here, into the sieve tube element. And what is the effect? What is the impact of this happening? It lowers the water potential, which we can sometimes symbolize like that with a Greek letter psi. Why? Whenever we have the addition of a solute, that will lower the water potential. Remember, concentration of solute and water potential are opposite to each other. So that lowers the water potential in here. So what does lower potential do? What is going to be the consequence of that? So number two, the consequence of that is that if you've got low water potential in here, you're going to tend to want to draw water in. Where is this water going to come from? It's going to come from the xylem. That's the closest source of water here. So here, this is the xylem. This is my phloem, my sieve tube elements, right? Xylem is transporting water up from roots to leaves. So number two is that water is going to be drawn in from the xylem and enters the phloem by osmosis, as always. Water moving is always osmosis. Thereby increasing, so what is the effect now of water being drawn in? More fluid. It increases the hydrostatic pressure. Again, this idea of hydrostatic pressure, like we saw with the blood vessels. More fluid, more pressure of the fluid against its container. So it increases the hydrostatic pressure. Pressure in here, I hope you can see that, HP. It increases the hydrostatic pressure. Number three, step number three, let me just change color. Step number three is going to be that so now you have an increase of hydrostatic pressure up here, which will basically push, it's going to create a net push of the water plus assimilates the sap basically downwards. So there's going to be a big push downwards. When the assimilates now reach a region which has, and of course, here is my source, right? And here's my sink, which is typically the root. When it reaches here near the region of a sink, the assimilates are going to be unloaded, into the sink. So number three here is that the assimilates are loaded into the sink. What is that going to do? It decreases the water potential in the phloem. So therefore, some water, let me just do yet another color. Let's do purple. So water is also going to leave the phloem. So number four, water is also going to leave the phloem and go into the sink. Why? Because when the assimilates got moved into the sink, got unloaded into the sink, what did that do for the sink? It lowered its water potential. So water needs to be drawn in. Where is that water going to come from? It's going to come from the phloem again. So now the hydrostatic pressure, number four, now the hydrostatic pressure, let me just do purple again, sorry. The hydrostatic pressure now is decreased. Compare that to that. And finally, this entire flow creates a hydrostatic pressure difference or gradient, you could also say, which pushes sap to the location that is required by mass flow. So basically this here is my, let me just choose another color. Let's do orange. Hopefully those are different enough. So step number 5 basically refers to this net hydrostatic pressure that is created. Just going to give you like 2 seconds to copy down the annotation on the diagram If you can use different colors, try. If you don't have them handy to yourselves now, just kind of like correlate the numbers to the part of the diagram, and then later you can fill it in with colors. Take a picture of the screen if you want. It's probably faster. And we are now going to move to a example question. Plants have developed a mechanism to ensure that assimilates are transported to where they are most needed. Describe how assimilates are transported through the plant. So 4 marks. So our context here is the translocation, the phloem and mass flow. Our command word is describe, and our guide, our direction is that we are dealing with only assimilates and not water. Take a minute, just a quick minute to try to structure the answer for yourselves. So again, this is one of the questions with what we tend to call a generous mark scheme, which basically means that there are more marks available to mention than the 4 marks that are needed in the question. Let's go through them. So again, it's a describe. We don't need to give a lot of reasons for it. We just need to be as detailed as we can, making sure that we cover at least 4 marks. Just a super quick advice that especially if you're going, for really, really high marks, it's often not a bad idea to write, like, don't just say, oh, I wrote 4 marks, fine, I can stop now. Try to aim for writing at least 1 more mark, because you never know, sometimes you might have not phrased something exactly as it should be. So having that extra mark there is going to ensure that you're not going to lose any marks. So assimilates are loaded into the sieve tube elements of the phloem at the source, mark number 1. Just do it right, so it's a bit more obvious. What does that do? What is the effect of that? It decreases the water potential of the sieve tube elements and decreases the water potential. That's 1 mark. And what's the consequence of that? Water from the xylem moves into the phloem by osmosis. That's another mark. Thereby, what's the result of that? Increases the hydrostatic pressure of the phloem at the source. It's really important to, state the places where everything happens, to be precise. Now, at the sink, the assimilates are unloaded and water follows, thereby decreasing the hydrostatic pressure. The fact that I've got increased hydrostatic pressure at the source and decreased hydrostatic pressure at the sink, means that this difference in pressure between source and sink transports assimilates by mass flow. This difference here also gets a mark. So it's basically the net difference in pressure, in hydrostatic pressure between source and sink that drives mass flow. Let's go to the very last bit for today, which is the experimental evidence to support the theory of mass flow, or the hypothesis of mass flow. And we're also going to do the cohesion tension theory. So, what are the scientists? What are the evidence? Companion cells contain many mitochondria, so they just saw that by observing them by the microscope, with translocation stopping when mitochondria are poisoned. So they did an experiment where they inhibited the function of mitochondria, and they saw that translocation stopped, indicating that it's an active process, first of all. Secondly, the rate of flow of assimilates in the phloem is much faster than could occur by diffusion. So again, suggesting that cells actively load and unload assimilates at the source and at the sink and the source. Finally, if you cut the phloem, as this is showing here, so this is my phloem. Oops, phloem. This is a little knife that I can use to cut, and sap is released under pressure. So you can see that the liquid in there must have been under pressure. If you're OCR and Edexcel, don't worry about this one, Edexcel A. What is now the evidence for the cohesion tension theory? So this is what I told you before about this idea of the negative pressure. What does that mean? And also, how do we kind of interpret it in graph form? The mass flow hypothesis concerns translocation. The cohesion tension theory concerns transpiration. So don't mix those up. So this is the xylem, transpiration. Xylem. So what we can observe is that the diameter of tree trunks, entire tree trunks, will decrease in the day. This might be really subtle, but scientists were able to measure it with sort of accurate equipment. Will decrease in the daytime and increase at night. So basically, it shrinks in the daytime and it relaxes again, it expands again at nighttime. This suggests that there is negative pressure that increases when water flow is the highest. You can think of that as, like if you're, for example, drinking something with a straw and you're sucking, you're creating negative pressure inside the straw. That's like as a way to understand that. So this here is my branch diameter in red, and this here on the x-axis I've got like hours in the day. So 12 is 12 noon, when the sun light is the most intense. What does that mean? The sunlight is at its most intense. Photosynthetic rate is at its highest. Stomata are open all the time to facilitate gas exchange. So therefore, the amount of transpiration of water from the leaves is the highest, which will also mean that the transpiration rate is its highest. And that will coincide with the lowest diameter, branch diameter. So I've got an inverse relationship here. What is further evidence that supports this? If we cut xylem vessels and they basically draw air into the xylem rather than water dripping out, which is the opposite to what was happening in the phloem. If we cut it, sap basically kind of squirts out, indicating that it was under positive pressure, pushing against the walls. Here, air is sucked in rather than water coming out, indicating that it was under negative pressure, inward pressure. If a single air bubble enters the xylem, it prevents any water from being carried into the vessel. So again, indicating the need for a single unbroken column of water. Let's look at a final exemplar question for today. A scientist was investigating the mechanism involved in loading sucrose into the sieve tube element. They isolated the companion cells and conducted some experiments, making the following observations. Observation number 1, companion cells became negatively charged compared to their surroundings. Number 2, the pH within the companion cells increased Well, the pH of the surrounding solution decreased. 3, addition of cyanide, which is a poison, prevented the change in pH occurring. What conclusions can be drawn from these observations? So, context here. The entire explanation of what is going on, the mechanism of active bloating, command conclusions, which basically effectively means to analyze and evaluate the data, and direction, that I need to use the data, the observations only here can be drawn from these observations. Okay. So we're going to go through the answer together because we've run out of time. The positively charged hydrogen ions are being, so what do we notice? First of all, we notice that we're talking about pH. What is the first connection that I have to make in my head? What is pH? Again, I have to remember my GCSE chemistry, right? Even for those of you that are not taking A-level chemistry. What is pH? It's a measure of hydrogen ion concentration, isn't it? Hydrogen ion concentration, basically. So decrease in pH means more hydrogen ions. Increase in pH means less hydrogen ions. So it can be concluded that the positively charged hydrogen ions are being moved out of the companion cells into the surrounding solution, which is why I've got, where is it? pH within is increased, pH outside is decreased. Increasing the pH inside the companion cells. Sorry, there's a little bit of a mistake here, actually. Positively charged hydrogen ions are being moved into, out of the companion cells into the surrounding solution. Increasing, sorry, my bad. Increasing, sorry, it's the end of the day. Increasing the pH inside the companion cells, less hydrogen, lower pH, higher pH. This process requires active transport, as addition of cyanide will block its progress. Second mark. Cyanide, and explaining why cyanide does that, because it prevents the production of ATP by blocking effectively mitochondrial function. Specifically, we're saying that it blocks the electron transport chain. Don't worry about that too much if you're year 12 and you haven't done the details of the reactions of respiration yet. If you said that it blocks mitochondrial function and production of ATP, you would still be fine to get that mark. Okay. So thank you very much, everyone, for your participation today. I'm just going to put up the screen again for the survey. Sorry, hang on a sec. Where is it? So you guys can have a look. If I can find it here. Just in case you didn't have a chance to copy it down. But that's it from me today. Oh, and it's been sent yet again in the chat with the correct, with biology instead of chemistry. So just copy it from the chat. And I'll be seeing some of you tomorrow. Those of you that are finishing today, congratulations on making it. And best of luck, of course, in your exams, in your June exam or your final exam. Bye.